This is my video for the Unit 6 test correction. So I chose the prompt from 2009, Form B, question 1, and the question, the statement was, describe how plasmid can be genetically modified to include a piece of foreign DNA that alters the phenotype of bacterial cells transformed with the modified plasmid, and then describe a procedure to basically see how, if it was successful, the modification. So basically, the way to understand for this question is what a plasmid is, how they are used and what they're used for, what techniques are used to insert the genes or the gene wanted, basically, and then what the desired goal is and how to know if you achieved your goal. So for the first part of the question, basically you have to answer is a plasmid vector should be used, which will basically carry the inserted gene. And what a plasmid vector is it's basically a circle of DNA. And I have a picture that will show that later. So the plasmid is basically going to be cut open, or the scientific term, I guess, is cleaved. But it's going to be cut open at a certain location to allow for the desired gene to be put in. And then the desired gene is going to be cut. And what's important to know is that you're going to use a restriction enzyme, which cuts specifically at one point for the gene. And then you have to use that same restriction enzyme to cut open the plasmid. And then once you have both parts cut, you're going to put the gene in. But it's important that when cutting it, that it was cut so that there would be sticky ends, which is basically the overhanging of the nucleotides, which I'll show in the next slide. And once you have that, it's almost like a puzzle, so it allows for easier matching. And then once you put the gene in, you're gonna have ligase, which will basically seal up the plasmid. And if you had used something like blunt ends, you wouldn't be able to really put the desired gene into the plasmid, because what the blunt ends do are, it doesn't really allow for that puzzle to connect per se, because it's basically just, straight cuts. It's not really specifically cut. So what the picture shows is you have basically this whole thing is the plasmid. This, you would say, is the desired gene. And notice how it's not cut basically straight. It's cut so that's like straight up straight. And that's basically what a sticky end is. But if it was cut straight, that would be a blunt end. So when you have like that, it makes more sense that it would fit in like a puzzle piece. And then what's used to cut here, like I said earlier, is a restriction enzyme. It's going to cut there, and you have to use the same restriction enzyme to cut there and cut there on the plasmid. And then this will be your final product once you have the inserted gene into the plasmid. So the next part of the question is how to know if you were successful. So basically, once you have the plasmid inserted, or once the plasmid is ready, you're gonna insert it into the cell, and you can insert it via transformation, which is when the bacteria itself takes up the new DNA. And to see whether or not you're successful in expressing that desired gene, you would obviously look for physical traits, which is basically what gene expression is. So the reason for the gene, like if you did not see the gene, or if you did or did not see the gene that you were looking for was expressed or not expressed, that could be reasons such as pH level in the cell, temperature, the amount of nutrients, or the supply of ribosomes in the cell. And then basically to summarize, well, now I have a better understanding of inserting the gene, but something I wish I had a better understanding of was labeling the plasmid, such as here, because I know for 1 to 3 responses we had to label specifically where we would cut and just different factors of that, and I wish we had a little bit more practice with that. But other than that, I think I have a pretty good grasp on inserting genes now, and there's my works cited.